Hello, Tom.
Hey, how you doing, Bradley?
All right, how are you?
Good.
Long
How
time
you
no
been
see.
doing?
What was that?
How are you been doing?
Oh, pretty good, pretty good.
That's good.
How about
Oh,
yourself?
that was loud.
Uh, Here's Valentina. Hello,
Hi,
Valentina.
hi.
Hello, how are you?
I'm fine. You?
Pretty good.
Okay, great.
Okay. So, welcome to the meeting. If the other people come in, we can catch them up on things. It's the second meeting of the year. So last week we had the first meeting, um, and so I have a bunch of things to talk about today. But Tom, I know that you Dick alerted you to the 1D Ising project that had
Yeah, was
been.
he gonna be here?
I don't know.
Okay. All right. Well, I'll catch up with him at some point.
Yeah.
I was trying to read up on that.
Oh, so what is it?
I'm not sure yet.
Oh, okay.
It's one of the like the patterns, the Wolfram patterns. I think he's got an idea. I was just going to revisit that kind of.
Okay. Yeah, that sounds interesting. Yeah, revisit it with some modern techniques or with, I guess,
Yeah,
Wolfram's
well,
been doing
I
that.
think it's related to like the seashell patterns and the shell patterns. Like
Okay.
they are the cellular automaton things that some kind of thing related to that.
Yeah, yeah. It's I actually have something interesting on that this week, but
Oh,
wait
dude,
for that.
okay, cool.
Yeah, it's a paper I found from 1986. So the computer science is a little bit dated, but it'll be interesting. I'm sure it'll follow it. I know Dick has been interested in the seashell patterns for a long time,
Oh yeah, yeah.
and yeah.
Yes,
So,
it is.
oh, what was that?
Yep. Oh, he certainly is. Yeah.
Yeah, yeah. So, Valentina, anything new going on with you, or?
Uh, yeah, I'm working on a different project now, but mainly on a unified structure.
Okay.
I don't know. Maybe I can share some of my work next for the next meeting.
Yeah, that'd be great.
Oh yeah.
Yeah.
There are some question that yeah, I need to work on actually.
Yeah. Yeah. So yeah, let me know when you want to present, and we can find time during the meeting, and we can do that. Okay.
Yeah, maybe you can help me in my project too.
Yeah.
Because you are very expert in image analysis, and maybe yeah, maybe you can suggest something.
Yeah. Yeah. I'd be happy
Thank
to.
you.
Yeah.
Thank you.
Okay. So coming up, we have a couple things. First of all, we have the Google Summer of Code program, and last week I talked about some of the projects that we'd be doing this year. And so let me share my screen. This is the thing I submitted to the INCF. So we had two projects. I think I mentioned this before. The graph neural networks as developmental networks, and then the digital microsphere, which is the stuff that Susan's working on with the axolotl data. So the first one, of course, is the graph neural networks as developmental networks. I don't know if I had written the whole thing up by last week. I think I may have. But again, to mention this to people who are here, so we're looking at different types of networks in biology and biological development. So there are things like neural connectomes, gene regulatory networks, interactome networks, and anatomical networks. And the idea is we have these cell tracking data that we've been working with. We've also created cell tracking data by segmenting cells out of images, and then creating databases with that. We've done that with deep learning, different deep learning techniques. The last several summers, we've done a number of different deep learning algorithms, and we have the DevoWorm package now, which is, now there's a preprint on it, which is here, and it describes the platform, and it's on PyP, so it's open source. But the idea here is to extend that library of things. So we have things in DevoLearn, and we have like these different deep learning techniques. And then we also have this DevoWorm AI, which is a platform where we have different tools that you can use to extract data out of an embryo, basically. We haven't benchmarked it on anything more than embryos, but we've looked at mainly C. elegans embryos, and there are a couple other types of embryos and developmental cell type images where you have cells that are dividing and differentiating. So those are all sort of, those platforms would help us do those things. The graph neural networks idea is to extend that to sort of creating these graph embeddings, which actually are representations of some of these networks. So we're extracting data from images, or we're using cell tracking data, which is where they put a dye in the, in the cell nucleus, and then they track it with computer vision. So, this can be done either as you're collecting the data or as in the form of secondhand data. But in any case, we can use these data to create these graph embeddings, and then we can actually use those as sort of like templates for different graphs. So if we want to describe, for example, the C. elegans connectome as it's developing, we can, use these the first class of models, the pre-trained deep learning models, which those work by just taking some data and developing the algorithm so that they're sort of well adapted to that data, and then you use that. You don't have to use a training set anymore. You have this. The training data is embedded in the structure of the algorithm. So you just take the algorithm and you use it. And you don't have to train it necessarily. That's limiting in a number of ways, because as you can imagine, if you have a lot of out of distribution data or other things like that, it makes it hard to adapt the model. So we have a pre-trained deep learning model for C. elegans. If we were to apply that to like Drosophila or some other embryo, like a spider embryo, we wouldn't be able to do that very easily. With graph neural networks, you basically characterize the network embedding, and then you use these neural networks to analyze the data. And it's just another way to like extract data and make some, basically say something about the biological system. So this is a newer technique, graph neural networks. We've talked about this in meetings before. And I'm just looking for people to work on this library. This is going to be I think, one of the shorter, so they have two different types of project. They have the 175-hour and the 350-hour project. So this is a shorter project, although I'm not sure if maybe it should be a longer project. I don't really know how. I don't know when I have to make that. We have to make that decision on this, because it's hard to know. I guess it'll depend on people who apply, and if they think it's something that they can do in that short period of time. All we're looking for is just for people to develop these graph embeddings and test them out a little bit. But we'll see. I think that should attract people. Every time we offer something with machine learning or deep learning or anything like that, we get 20 applicants. So, and then the digital microsphere, which is this, where Susan has her ball microscope that has all the different microscopes sort of embedded in different locations around a microscopic plate. And then you have the embryo within the plate, and it's able to measure, get captured, like 9 or so different perspectives of the embryo. So, you're looking at it from the bottom, from the top, from the sides, from diagonally, and then you have a series of images from each of those perspectives. So then what you can do is you can stitch those together into these spherical representations. So I have 9 images from different perspectives, and they get like probably most of the surface of the embryo. And so then what you can do is you can take each of those perspective views, tile them onto a sphere, and then use different warping algorithms to fit them onto the surface of the sphere. We had some people working on this last year, but no one got selected for the Summer of Code project. And I mean, we have a couple people, I don't know if they're going to reapply, but it requires you to have a good handle on these sort of mathematical models of warping and implementing different techniques for building maps. So, this is like the surface of the Earth. It's like you have, we have flat maps that are basically taken from things that are on the surface of the Earth. So, a map of the world is basically taking the surface of a sphere, pulling it off, and then flattening it out and using a projection to approximate some of the gaps that you're going to find. And so this is the reverse of that. This is where you're taking a flat image that's captured as flat image, I should say, and then you're tiling a sphere. And so that's most of the technical work. And then Susan will provide a lot of the data. She's got more data coming. We have probably at the present, I think we have several gigabytes of data, but we would like to have many gigabytes of data. Probably not terabytes, but I think for a summer code project, that's probably enough. And so just once we have this platform and once we have the software, we can just keep generating data and keep creating or modifying the atlas. So people are interested in applying up to that. That's what's going to be up on the INCF website soon. And so the project schedule is that we'll have these applications up for a while. People will be able to apply to them, and then we'll have the application process where people come up with a solution. They can attend the meetings here, get feedback, and then I think I don't know when the application period formally opens, like in March or something, and then they have a month to put their applications in, then we evaluate them. Then sometime in May, the selections are made. So that's always fun, because people are always like excited about it, and then we only get to pick we only get a couple slots per year, so it'll be, but people will be able to interact with the group here, and they could stick around and pursue the project even if they don't get selected. So, I think that's probably enough projects for 1 year. Last year we had 3 projects, and we only got 1 project selected. So, that was that's coming up. So the next thing I'd like to talk about is, let's see. So Dick sent me this article, and I thought it was interesting. This is Earth Sky. This is a popular magazine. And the question is here, can we and should we send life to the nearest star? So this is a nice speculative piece. Dick has an interest in astrobiology, and I'm interested in it too. I've had a long-standing interest. I never really pursued the science of it. But we have this article, and this is a tardigrade. So this is an organism that...
Extremophiles, right? We have this concept of sending alien life, to outer planets.
Yeah.
Yeah, it's pretty exciting.
Right, yeah. So this is what they call a tardigrade. And tardigrades are very resilient organisms. They can survive the vacuum of deep space, apparently. And they can, they've taken them up on the space shuttle and various things. So they know they're pretty resilient. And the idea is to send these tardigrades into outer space and things. And they're about 1 millimeter long. So they're maybe at about the scale of C. elegans. So, imagine this. You can't see this like this. You'd have to look under a microscope to see a tardigrade. But nevertheless, they're very resilient, and they can be sent to to another, and on a long-term mission without having to undergo any sort of life support or anything like that.
Even I was reading, a similar article based on tardigrades sending them into space. The only problem that they were facing, was that these extremophiles, especially tardigrades, they only reproduce when the conditions are exactly right. That's why, they only found maybe beneath the tree moss, so like there's a moss layer on trees, right? So they have like specific conditions where these kinds of creatures thrive. So they'll be able to survive the harsh conditions, that is not an issue, but I think that article clearly mentioned like we are still a very long way, from sending them to space. There is, they're barely nothing to do with and all that.
Yeah.
Things that they find easily as well.
Yeah, I think it's like I said, it's a speculative piece, and they have, they're sort of identifying the organisms that they might want to do, and they have to be what Karan called extremophiles, which is organisms that can survive extreme conditions. So we have these tardigrades, but we also have C. elegans, and that's our target organism in this group. So nematodes actually are well known for being able to survive in frozen conditions. So they've found C. elegans eggs in 40,000-year peat, 40,000-year-old peat, where it's been at 0 degrees Celsius or below 0 degrees Celsius for many years. And so they've been sort of sitting in there. And so basically the egg is just this, container containing DNA and some cells. And, I guess it can survive for that long without degrading. What they basically do to revive a colony of C. elegans is they have the eggs, they're frozen, and then they just put them in a plate with some food, and they end up hatching and then starting up. You can freeze C. elegans pretty easily. You just have to basically extract the eggs from a population of worms, and then you can freeze the eggs in a medium, and the eggs will be, remain intact, so that when you unfreeze it and you put it on medium, they just start to, they end up hatching and going through their life cycle. So C. elegans is another candidate. I think it's kind of interesting, some of these organisms, maybe it's a nice sort of thought experiment to think of all the organisms that you could say put in that class of organisms that you want to send to space. What makes them sort of candidates for that? And so I would imagine like one of the criterion would be you have some way to, so they can survive deep freeze of some type. So, they might have eggs that are very resilient, or they might be able to, have an outer shell of some type that they can, hide in. They might have a large population size, so you can get a large population from a very small starting population. So, you only maybe have a few eggs. They can, proliferate into maybe thousands or hundreds of thousands of organisms over, a period of time. So you can have these very small, what they call founder populations, and they can become very large. I mean, these are all candidates that you would want. And so, there are probably other examples you have, for example, different types of archaebacteria. There are other types of extremophiles that might fit this bill. So the scientists want to know, what would it take to send life to the nearest star? So they're targeting Proxima Centauri, which is about 4 light years away, which is one of our nearest neighbors in the cosmos. And this is an area they call experimental cosmology, which is the idea of using the whole universe as a sort of laboratory to test ideas in physics. And so they need to find technology for it, obviously, like some sort of ship that would go there in a reasonable amount of time. You can't spend like 10 million years because no one will be here to make, I don't know if we'll still be around as a species, but it's not likely that anyone will remember it. So now you have to create a ship that would get there, in a reasonable amount of time, like within a century maybe. And so they're having to any ship that would do that would have to travel at a pretty high rate of speed. So 20 to 30% the speed of light, maybe. And then this kind of goes through some of the things, but this is nematodes. So they talk about nematodes being able to go through, not only have like these eggs that survive freezing, but also that they can, there these periods of suspended animation. So that's good.
Did they discuss the ethical aspect of this?
Let's see.
Because, I mean, some extraterrestrial civilization sent little bits of life to us and landed on Earth. And they
Yeah.
caused a catastrophe.
Yeah. Yeah, here's the interplanetary ethics section here. So other scientists involved in this study included a radiation specialist and a science-trained theologian. And I think what Tom is saying is that, you send like some sort of organism to another, and it lands on another planet. It can really wreak havoc on the planet, especially if there's life there. It can wipe out, like, because if you imagine, could have like some sort of alien bacteria or virus, land on Earth, it could do a lot of damage here with our.
Can you hear me?
What's that?
Can you guys hear me?
Yeah, you're pretty low, but
I just want to say we just published a book on astrobiology ethics.
okay.
Goes into this stuff in very detail. Maybe I'll see if I can find it.
Yeah, that'd be great. So yeah, they're. I guess. Well, Dick has. They have
Yeah, I
a
was
book
just
on that.
wondering about that.
Yeah. And that's of course panspermia, which is what I think. Where life goes from one planet to
We don't go
another.
into ethics there. Here it is. Okay, got it.
Yeah.
Okay. I'll put the link in the chat. Where's the chat? Open chat.
There it is.
Okay.
So this is on astrobiology, science ethics, and public policy. And so this is it covers a wide range of things.
Yeah, it includes these questions.
Yeah.
There's also one in the panspermia book saying it's too late in terms of Earth and Mars. And the reason being that Earth and Mars have been exchanging meteorites for 4 billion years.
Oh yeah.
Okay, and therefore, if we get to Mars, we're probably going to find Earth creatures.
Oh yeah.
Right.
That's what I'm expecting.
Well, if we want to actually tell other civilizations about life, we could always send dead water bears, right? So they don't destroy their biosphere. Like they can look at the DNA and stuff,
Yeah.
without actually having water bears, engulfing everything.
Right.
When we get there, we'll find water bears all over the place, like water bears building skyscrapers and stuff like that.
Yeah.
Like termites, huh?
Yeah.
Oh, you never know.
So, Dick, Tom and I had a question. We were talking about the Wendy Ising project. And,
Okay.
yeah.
Let's see if I can show you a picture. What brought this thing up in the first place? Let's see. Just a second. I sent it to myself, so I'm getting up. I don't know what's going on. Something's going on with my mail, or I can't see.
Oh.
It's coming up in a weird format. Okay, let me just describe it. With Shruti and Krishna, we did a simulation of the one-dimensional motion what I call wraith factor. Big molecules in the wraith of the diatom. We assumed it was one-dimensional. We assumed there were interactions between them and whatnot, and it basically became a non-steady, or a sort of a steady state Ising model instead of equilibrium. And we were trying to come up with this visualizing it. And what we did is just made consecutive states to draw them. Bradley, I think it's in your mail.
Okay.
Perhaps you can open it.
I can't really get to it right now, but I think I've seen this before. It's like a straight line with different.
Yeah, it's a straight line, but if you repeat the straight line, which changes with time, you get something that looks like a Wolfram pattern.
Oh, okay. So it's like the one-dimensional automata.
Yeah, it's one-dimensional, exactly. So one-dimensional automata, and the other axis is time, which is the same in the Wolfram patterns. They're the coordinate down the page is time.
Okay.
Okay?
Yeah. So let me see. I can share my screen. I can actually do this on a board here.
Okay. And then skip to the figures.
Yeah.
Just do a search on the figure.
Oh, well, I don't. Yeah, I can't get to my email here, but I'll just do this. Oh. Hard for me to get this to draw here.
Okay.
Yeah. Anyways.
At any rate, what we did then is repeated the pattern as it developed over time, and it produces something that looks like a chaotic Wolfram pattern.
Okay.
This is what then brought up the possibility of generalizing this to all Ising models, and unfortunately there are 800 papers on them. Okay. One-dimensional, not one of which mentions Wolfram.
Oh.
Okay.
Yeah.
So, there. Valentina, are you familiar with the shell patterns on some snails?
No, I'm not familiar with
You're
this.
not familiar with those.
No.
You have Wolfram patterns. You're going to have to read Wolfram's book, which is an enormous tome. It's a huge book in which he slowly reduces mentioning his own name only 200 times per page.
Huh.
Okay.
Oh,
I think there's a little egomania there, but
Yeah. Yeah. Maybe.
And never call it Steve. He's Steven.
never called Steve. Okay.
No, someone did that at a conference. They said they introduced him as Steve Wolfram and he got upset.
Okay. Any rate, if you take an Ising, a one-dimensional Ising model, and repeat it over time, you get something that looks Wolfram. Now this brought us to interesting questions like, okay, are the patterns similar to any of the Wolfram patterns? That'd be one question. And then we end up with a much deeper question, and that is, are Wolfram patterns closed with respect to patterns? In other words, if you change the rules, you end up still getting a Wolfram pattern, or you increase the space of patterns.
Well, he enumerated all the patterns that are like with neighbors of one or something like that, right?
Yes, exactly.
Yeah.
What I'm suggesting is we generalize that to a larger neighborhood with more rules, and the question is, are those rules redundant, or do they generate new patterns? And that's how, now, the reason that's important is if it's closed with respect to the original Wolfram rules, notice that the original Wolfram rules generate all the patterns you can get. Then Wolfram's new kind of science, which is the name of his book, his enormous book, would have some basis, possible basis. But if it's not closed, then there are things that could happen even in his universe which are not part of his rules. Okay? So that's why I think the question of whether Wolfram patterns are closed or not is perhaps the best way to start with this. And one can add, you could add, for example, just one pixel, you know, he does the left, the right, and the previous thing in order to generate his rules. You could add 1 or 2 pixels. I mean, you want to pull up a Wolfram pattern so Valentina knows what we're talking about?
Okay, so this is an example of a 1D automata. I drew this on my board here. So let's go over this. So this is a one-dimensional automata, so the one dimension is this line here. And as you can see that you have several cells that are sort of next to one another. There isn't a second dimension where you would have rules. In the up or down position, it's just right to left. So you can see that this automata has 2 states, on and off. The states are then determined by these neighbors. So this cell here has 2 neighbors, this one and this one. This is a neighborhood of order 1, so there are 3 cells in this neighborhood. And the focal cell is here, the neighbors are here, and then the rules basically define how this cell is turned on and off. So if this cell is on, then this cell is on, and if this cell is off, it's still on, because the rule here is that if either cell is on, then this cell is on. And so each cell can have different rules. And so we'll be talking in this meeting about how, you know, does the automata do things beyond the rules if you model, say, like a biological system, or does a biological system, if you model it as a 1D automata, behave well within the scope of all possible rules that are applied to it?
Okay. Let's see. Yeah, I'll tell you what's quickest here. Let's see. But visit this one. Okay, here, I'll put a URL. It's probably quickest.
Okay. Okay. So this is, oh my gosh, it's covered in Rule 30s. Okay, so this is an example here of a 2-dimensional.
Okay, now Valentina, you got an assignment. Look up patterns on snails.
But it's like the pattern on gastropods.
Yeah.
Yes, gastropods,
Yes.
snails.
Okay.
Yes, they make patterns like these, and people presume that this mechanism is working.
Okay.
Okay. I'm not sure it's right, but one of our projects is to eventually test that.
Yeah.
Mm-hmm.
So this is, yeah, these are the,
I have a book. Just me. This one.
What's that one?
oh,
The algorithm. Yes,
Yeah.
yes, it's in there. Yes, it should be in there. I used to have that book. Yeah.
Okay.
Okay.
yeah.
Yes. You'll find patterns, all sorts of patterns in snail shells, and the question is, can they all be subsumed under Wolfram rules, or are Wolfram rules not closed? Okay, so I think we can ask a very fundamental question about the Wolfram pattern. Wolfram would like to claim that the basis of all physics.
Mm-hmm.
Okay. Okay, if you look up his books, his big one is on the new physics, which he's pushing. Okay. So anyway, he might not be happy with this, depending on the results. Okay, so
I thought later on he's like, he sort of drifted off to, it's not, you know, all physics is not a cellular automaton, it's some kind of graph. Like,
Oh.
the universe is a giant graph now.
he's got a little away from these, but still,
Okay, yeah, right.
it's worth looking at. Is how closed are these? Now, one of the things that came up with is take any pattern here that he generates, and for each pixel substitute a picture.
Okay.
Okay? Have you ever seen these things, pictures, where the pixels are pictures?
Yeah.
Yeah. Well, we can do it with this. Now, are those subsumed under Wolfram rules or not? I'm not sure. I think we have to think that thing through.
Yeah.
Okay. And basically, you just assign a binary number to each of the pictures you use, and then you get the same rules except each pixel would be a picture. Okay?
Yeah.
So I think there's some fun things to do here.
Yeah.
Okay?
Yeah.
So this is what it came out of the one-dimensional slit in the diatoms. One's called the wraith, and our attempt to model it, and then our attempt to show people what we got. And that's what led to the whole idea.
Yeah.
Okay?
Mm-hmm. Sounds interesting.
Yeah.
Okay?
Yeah.
Yeah.
So have you got some data for that, or?
No, I haven't done anything with it.
Okay.
Now, this comes out of my high school days in a way, when I first learned about imaginary numbers. Okay? And imaginary numbers are constructed by, I times a number plus another real number. Okay? And I was a smart ass, and I figured out, okay, if people invent imaginary numbers, I can take imaginary numbers and invent the next level. Okay? So I did that, but then I found out that the higher levels of imaginary numbers were still imaginary numbers, and therefore imaginary numbers are closed under those operations.
Yeah.
Okay?
Cool.
Okay. So my attempt to generalize that in imaginary numbers went nowhere.
That's great. Yeah.
Okay. So that's over almost 60 years ago.
My older sister once came home from school and claimed there were negative numbers, and I said she was a liar.
Oh.
They can't possibly exist.
Anyway, so I think the question of what does closed mean for patterns, and are the Wolfram rules closed or not, as far as I know, has not been addressed in the literature.
Huh. Okay.
Okay?
Yeah.
So we might actually make a contribution to mathematics.
Yeah.
Okay?
Okay. Well,
Okay, so, I leave it at that. I'm going to let you guys mull it over and think of how to approach it. I mean, we could just go to the Ising literature and simulate Ising models, which might give us some ideas, and it may or may not give us new patterns.
right.
And how do you judge if a pattern is new, especially if it's chaotic?
Yeah.
Okay, and some of the Wolfram rules do generate chaotic patterns. So now there's a small literature on what are called stochastic Wolfram patterns, where the rules have some probability thrown in.
Right.
Okay, so I got that far in terms of literature searching. But again, I don't know about the question of closure.
Yeah.
Well, if you remember what we did with Morphozoic, it might have some relevance here, because it actually looks for like increasingly sized neighborhoods back in time, to encode neighborhoods,
Right.
So we could kind of think about that.
Yeah, the other thing to think about is I'm not sure whether that came out of this or not. Yeah. The adaptive neighborhood.
It's a morphogenesis kind of thing,
Yeah. Do you know what an adaptive neighborhood is?
Maybe. Go ahead and tell me.
Okay. Consider a 3 by 3 square of pixels. Okay? You can define a contrast between the central pixel and the one surrounding it. Okay? Now multiply by 3. Consider a 9 by 9. Okay? And group them in groups of 3. So we can do a contrast between the ones around and the ones in the middle, the average values. And then you can go higher, you can do another factor of 3. Okay? So you can keep going up and up this way, and plot contrast versus size of the adaptive neighborhood. If you do that, you'll find that the contrast seems to have a peak at some point for a given picture. And the adaptive neighborhood, in a sense, finds the resolution of the picture.
Wow, neat.
Okay? Now, I did this in the 70s in regard to mammography, and it's taken off. It's got about 40, 50 papers, but okay? So it's now should be showing up in text, in the image processing textbooks here soon. So it's got a huge literature, and applying the adaptive neighborhoods to the Wolfram rules would be interesting, but it would increase the number of rules astronomically very fast. Okay? Unless we apply the rules to groups. Okay? The way I did for the adaptive neighborhoods. So I kind of backed off on that because the combinatorics gets bad fast, and that's why I said maybe we should just add 1 or 2 pixels. Okay, which increases, the number of rules he's got is what, 256, so you just multiply by a factor of 2 for each pixel added.
Well, there must be there must be a physical causation here in the diatom and the wraith, right? So it can't be doing magic, right? It's got to do some physics, right?
Yeah, but it's not making patterns.
What?
It's not making patterns. It leaves behind a slime trail. Patterns are just a way of representing the change in the state of the molecules over time.
Yeah.
Okay, yeah, go ahead.
That can't be like, infinitely complex. It's got to have some constraints, right? So what the chemistry is doing in there, right?
Yes and no. There certainly are constraints, but the possibilities are astronomical. So
okay. All right.
Okay. Hey, Bradley, you got that email from me, right? With paper?
Yeah. I can't open it.
Why don't you forward it to Tom?
Okay, I'll forward it to Tom. Yeah.
Okay. You can see what we're about. One of the figures points out that this is it's similar to a Wolfram pattern.
Yeah.
But basically, we're simulating a fancy one-dimensional Ising model, which is weird because it's steady state, it's not in equilibrium, and it results in motion of the diatom.
Yeah.
Okay?
Yeah.
Okay, that's all I got to say about it.
Okay. Well, you're in luck today because I have a thing that I found a paper that has a lot to do with seashell patterns and how they might be generated in seashells. So let's see. I'll share the screen here. So I was at Dynamics Days last weekend, and there were a number of speakers. And one of the speakers was this is the paper. Bart Ehrmantraut, who's a neuroscientist, a computational neuroscientist,
Okay.
and
George Oster.
And George Oster, yeah.
Yeah, I know. I met him.
Yeah.
30
So this,
years ago.
yeah. So this is a paper by these people, and this is from 1986 in, I don't know, the Veliger. It's a journal. I don't know if it's a journal anymore, but
Probably the snail journal.
yeah, maybe. So the title was A Model for Shell Patterns Based on Neural Activity. And so if you go back, you recall, of course, this is what a gastropod looks like, the shell. And so they generate these patterns, and these look like the Wolfram patterns. So in this article, they show these Wolfram patterns that get generated. That's like there's repetition down the column here, and it's generated by these rules, ostensibly. I mean, they match up to these rules, and you can do this on a computer. But then the question is, what is the organism actually generating as in terms of these rules?
Now, one of the things I learned about these snails is that the patterns seem to be generated by secretion.
Yeah.
Now, let me show you, just a second, I'll turn on the lights here. I'll show you a snail which makes this obvious.
All right.
Here.
Okay.
See this one?
Yeah.
You see the
Yeah.
lines spread out as the snail proceeds to grow.
Yeah.
Suggesting a simple growth model. These things secrete something, they secrete the pigment, and the pigment gets deposited, but as the snail grows, it gets whiter. Now, I don't know what that means at a cellular level, but it might mean that whatever's doing the secretion is getting bigger. Maybe it's more cells per secreting unit.
Right.
Okay? Okay?
Yeah.
So I've never seen this before.
Yeah.
Pick this up at a garage sale.
So I took out some figures. So what they argue in this paper is that these patterns are based on a neural circuit, and so they show these like stripings, and they actually do talk about how there's a secretion mechanism for creating some of these patterns during development.
Okay.
So there's this the anatomy of the mantle region. You have
Yeah, I
big
have
mantle
seen
edge
the paper.
gland. Yeah, and then they have in the paper here, they have some assumptions that underlie their model. So they're actually building. I don't know if it's a computational model, but it's a model that tries to describe this process. So they talk about the mantle and how there's secretions that are deposited on the shell surface, and then they kind of go through this model, and then they also talk about linking this to the nervous system of the mollusk. And so this is figure 9, so I think it's this figure. This is a diagram of the model where they have a mantle pigment cell, which is MPC, which are these here, these squares, these rectangular cells. PCN, which is a pigment cell neuron, which is this triangle down here. And so the PCN synapse is on the MPC. And then you get the MNN, which is the mantle neural net. So that's this underneath. So there's this neural network underneath. It's just in the case of a biological neural network, it's connectivity between these cells, and it's coming down to this CG or central ganglion. So a ganglion is just this cell that has a lot of reticulating connections, or a center of cells that has a set of reticulating connections, and they're usually used as like sometimes central pattern generators or other things. A lot of invertebrates have these ganglia, and so this is the model that they're using, and the idea is that they're doing this sort of it's producing this sort of pattern formation. It's kind of like a Turing model, where you have.
You know what it's similar to? It's similar to a visual receptive field.
Right. Yeah. That. Yeah, where you have inhibition on the lateral inhibition and.
Lateral inhibition. Right.
Yeah. So this is their basic model, and they're just basically saying that, it's the nervous system is made for this sort of, I don't know if you want to call it secretion management, but when you in the visual field, you have a lot of cells in the retina that are responding to some stimulus in the environment, and it has to detect patterns.
Do they relate this to the Wolfram patterns?
I don't think so, because this was 1986. I don't think Wolfram had. That big book that he has was published in 2002. He was doing work on it before then, but I don't think they cite him in here. Yeah.
Has anybody ever done a Fourier analysis on this? On shell patterns, like a 2D?
Not that I'm aware of.
Is that, what are the harmonics of that would go into decomposing the pattern,
Yeah.
By the way, for Valentina, let me point out something. Let's see. Let's load it up here just a second. Okay, now, if you take a shell, any shell at all, with any pattern on it at all, if we build a device for rotating the shell and just accept, we take a picture for each angle, but then we only accept the pixels that are in front. We can then unroll the shell. Okay, turn it into a flat pattern. It's very difficult to deal with a real shell because it's three-dimensional. So I'm saying that we could build a device for rapidly turning the shell into a two-dimensional pattern, and then once we got that picture, we can analyze it.
Okay.
Okay? So
Okay,
the
thanks.
trick is to only take the strip of pixels that are closest to the camera, okay,
Mhm.
for each angle, and then make a montage out of those.
Yeah.
Okay?
Okay.
And there are questions, of course, of what's the ideal angle. It may depend on the shell. It may depend on the resolution of the camera, things like that. So there's, some details to get into. Okay, now in regard to Tom's question, the question as to whether a paper like this is correct, one of the things I did in the 1970s, while I was still a young man running around the world, I'd go to snail shops and buy shells that were defective. And these were usually shells that had some trauma, and they had to regrow their shell. Okay, and then the pattern would resume after a little while. So this gives us a fresh start to the pattern at each time for each of these shells. Okay? Give me a second, I think I can find them.
Okay. So the rest of the paper is just like goes over those patterns, and they kind of generate patterns. So here they have this where they're reconstructing a pattern of a snail shell, but they're doing it with characters on this array. And so this is 1986, so they didn't do like a modern analysis of this or even a modern rendering of it, but this was basically they were reproducing the pattern using, I guess, ASCII 2 characters. So you can see the pattern here.
Oh, yeah.
Yeah.
When we printed it on paper.
Yeah, checkerboard pattern. Yeah. And all that.
Oh, yeah. I remember those days. Okay, here's an example. Okay, this shell obviously went through some trauma and had to reestablish its pattern. Okay? In fact, it might have had to do it twice. Okay?
Yeah.
So I've got a whole bunch of shells with that characteristic. Okay, that they're defective. We need a quick means of probably a robot for rotating the shell and taking pictures, maybe 360 pictures if you want every degree, and then making a montage out of the vertical strips from those pictures. And the device has to accommodate shells of all different sizes.
Yeah, I remember that. Yeah, and then I think I threw in like, why don't we put it in a mirror bowl, and just take it all at once. And I remember Steve was threw in on that, but I can't remember what it was. Maybe rest in peace,
Okay, that's an old. Okay, we've been throwing this idea around.
Yeah.
It's just that this connection with the Wendy Ising models and the closure kind of makes it more interesting.
Yeah.
Okay.
Hey, Bradley, in that paper today, are there actually nerve cells that are connected to pigment cells?
I don't think that's a direct connection. They actually do like.
Okay. They just like wanted a model and they didn't.
Well, I think the model was like where they had it. Pigment was produced.
The real snails have this thing here with the.
Yeah, I think so. Yeah.
They do.
Yeah.
So their nervous system is connected to the pigment cells.
Okay. Does that paper cite Waddington's paper?
Let's see. Yeah. So I think that there's an actual set of connections here. They're just modeling this like as a neuroscience you would model the different cells.
That's kind of interesting. So it doesn't kind of run as a. It's under the control of a nervous mechanism.
Yeah. Yeah,
It really
excitatory
is,
and
or-
inhibitory neurons. Yeah.
Oh, interesting. Yeah. So it's not just a kind of a chemical or a cellular.
No, it's actually
Yeah.
so.
So that may be worth following up in terms of relationships with Wolfram patterns.
Yeah.
Oh, interesting.
But see, this sort of suggests some kind of roughly top-down approach,
Right.
whereas
Yeah.
the Wolfram patterns are strictly local in terms
Right.
of the rules.
Yeah.
Right.
Exactly.
Now, they do cite Wolfram and they do cite Waddington and Cow in 1969. So they have.
Yeah, that's about right. Yeah, Waddington and yeah. The Waddington paper was the first one on these snail patterns.
Yeah, it's the computer simulation of a molluscan pigmentation pattern. And then the Wolfram paper was a cellular automata as models of complexity, which was, like, I guess, one of the first ones he wrote on this.
Probably, yeah. Before
Yeah.
I did the book, yeah. You can get it, by the way, you might be able to get the book online. I'm not sure.
Yeah.
But is this pattern functional as a camouflage or as other?
Usually it's covered up by algae. There are two questions there. Often it's covered up by algae, so you can't see it. And second,
Okay.
many of the snails repeat similar patterns on the inside surface.
Oh, wow.
Okay, so it's not obvious.
Okay.
Okay, and besides, many of these snails are what are called conus. They're of the genus Conus, and they're aggressive predators. They literally will drill through the holes of other shells and eat them.
Oh.
So I suspect they're usually pretty thick. I suspect the pattern has nothing to do with camouflage.
Wow. Okay. So the meaning is not clear. The functional meaning.
Meaning is not clear at all. Maybe it's a waste product. I don't know.
No, it's impossible.
You know, like you can tell deer have been by their droppings, so you know what droppings look like. Okay.
It will be great to identify functional meaning.
Yeah.
Yeah.
At any rate, it's an interesting problem. And one of the things is that I don't think anybody has critically compared the computer-generated patterns with real patterns. They just say, oh, they look the same. That's the same as saying that a pattern on a starfish looks like cracked mud because they're similar.
Yeah.
Okay. Is the pattern on a turtle? Is the pattern just the same as cracked mud? Dried and cracked mud. They look very similar.
Yeah, the hexagonal shape.
Yes.
Mhm.
Okay. So I mean, one of the problems in development has been this sort of thing. People think by analogy rather than say, is this the actual mechanism?
Mm.
Okay.
Yeah, it's the old cargo cult thing, right?
Cargo cult, what's that?
Have you ever heard of a cargo cult?
No.
I guess in World War II, some South Pacific islands, the Air Force landed and created bases and stuff like that, and they passed out all these goodies to the natives.
Right.
And then when they left, they figured like, well, let's build something that looks like an airplane, and then it will bring us some good stuff. So they made like airplanes out of coconuts and all that.
So now we'll go to this next set of papers, final set of papers for the day. Stochasticity, Determinism, and Self-Fate. I think that fits in well with our theme today. And this paper is on stochasticity and determinism and self-fate decisions. So we've talked a little bit about stochasticity, and it's basically where you have a model that's probabilistic and that is quasi-random, and that is it takes it basically generates states at random or probabilities at random instead of a deterministic system where you know what the outcome is going to be in advance. So, for example, flipping a coin is a stochastic process, and whereas flipping a light switch might be a deterministic process. So these are just, one small example, but in biological systems, sometimes cell differentiation works this way. In C. elegans, for example, we have deterministic cell differentiation. A cell from a certain lineage will become a certain type of cell. There isn't much ambiguity about that, maybe except in mutant genotypes. But a stochastic differentiation scheme is where a cell will be a stem cell and it can differentiate into a number of fates, and it depends on the environmental factors. It depends on the niche that these cells exist in. It depends on its neighbors, depends on the things that it's exposed to, and then that generates a certain type of cell at a certain probability. So that's the difference between stochasticity and deterministic, or stochastic and deterministic. So in this paper, they talk about this in the context of development. During development, cells need to make decisions about their fate in order to ensure that the correct numbers and types of cells are established at the correct time and place in the embryo. Such cell fate decisions are often classified as deterministic or stochastic. So if you think about this statement, ensuring that cells are established at the correct time and place in the embryo, that might suggest that this is a deterministic process. It's like the clockmaker model that people often adhere to in the Enlightenment and maybe a little bit later in the history of science. But actually, a lot of cells are stochastic, and a lot of differentiation is stochastic. And surprisingly or not, it tends to meet this criterion. Even when you have a lot of stochastic differentiation, embryos are not chaotic places necessarily. There's quite a bit of order. And so this is an interesting kind of, maybe paradox, maybe not, because if you know anything about chaotic systems, you know that chaotic things can exist on the edge of chaos, where you have a lot of stochasticity, but you also have a lot of order as well. So, however, although these terms are clearly defined in a mathematical sense, they are sometimes used ambiguously in biological contexts. And so I know that, a lot of times when biologists use the term stochastic, they're not thinking directly in terms of the mathematics, they're thinking in terms of some process that's probabilistic. And it gets a little messy because people just generally don't conceive of that underlying process very much. They just say it's stochastic, and sometimes that can be an excuse to say something is messy and I don't understand the mechanism. Or that the mechanism is hard to pin down. And so, there's a difference there. So we discussed these frameworks within which such clear definitions make sense and highlight when certain ambiguity prevails. As an example, we examine how these terms are used in studies of neuronal cell fate decisions and point out areas in which definitions and interpretations have changed and matured over time. We hope that this review will provide some clarification and inspire discussion on the use of terminology. So this is largely trying to clean up the terminology in the biological literature and link it more to the mathematics. And so we know that these two different types of developmental processes exist, deterministic and stochastic. And so we can look at this experimentally, empirically. They address these issues in this review. They give a mathematical definition of stochasticity and then discuss the potential origins of stochasticity in cellular and developmental systems. This is also then put into a biological context, which we resolve a biological phenomena, and this can influence the conclusions drawn. So this is going to then be shown to be the case in the central nervous system development. And so a mathematical notation of stochasticity is this mathematical model where you have a hypothetical experiment E or observation E with an outcome X. And so the outcome X is maybe the number of cells in a certain type that could be quantified as a particular developmental stage. So in other words, if you have an experiment and you have a manipulation, there's an outcome X. And so in some cases, you might have some observation where the number of cells of a certain type in a certain developmental stage varies between observation, although the underlying process is basically the same. So, we think of experiments as if you control them enough, they're they're reproducible, but in a stochastic system, sometimes they're not so reproducible. Sometimes you get fluctuations. And of course, we know this from the standard deviation of an experimental measurement. But sometimes this is more fundamental. This is actually the way the system works. And so you can see where this might be a problem if you're a hardcore experimentalist and you're trying to get a handle on the mechanism, that this fluctuation is your enemy, but in fact it's the way the system works. So the particular outcome the system attains is given by chance and is thus unknown a priori. So this is a problem for modelers because you have to sort of give these you have to make assumptions about the probabilities in advance. You can make, you can turn this into a stochastic or a randomly generated variable, but you have to know that this is the way the system behaves in advance. So formally, X can be described as a random variable, which assigns a number to each element in the event space. A random or stochastic process is a sequence of random variables that can be used to describe time-dependent stochastic phenomena. In mathematics, the terms random and stochastic are often used interchangeably. I might add that in a lot of biological contexts, people will use random and stochastic in ways that aren't mathematically sound. So when you say random, you mean that the values have shuffled around or that they're not they're within a range, but they're not predictable. Whereas random number generation in mathematics is a bit different than that. And the same holds true for stochastic. But we use these terms because they also have a popular definition and sort of a way to, sort of a shorthand to access this statistical space. Random variables and processes provide a useful means to describe biological phenomena with an uncertain outcome. So one way to think about this is by using a probability distribution function. And so your PDF, or your probability distribution function, you can think of this in terms of the classic bell curve, or what they call a random distribution. And so this random distribution is where you assign a probability px to each possible value of x. So your probability distribution has tails and it has a center of mass, and each point on x, which is deviation from the mean, which is the center of the mass of the distribution, and this is for a random distribution, it gives you a probability of each of those. So the tails occur at very low probabilities, and the center mass of the distribution occurs at very high probabilities. And so you can think of this in terms of outliers and central tendencies, which is the way people often think about this. Of course, this works for random distributions, which are normal, but sometimes you have skewed distributions, sometimes you have other types of distributions, exponential distributions, and other things like that. And so it's harder to make those predictions. You can use the different distributions as models, but you have to make that assumption in advance, and it changes the result in the underlying mechanism. So exponential distributions have much longer tails. There are a much wider range of values that occur at very small probabilities. When people say something has a long tail, that's what it means. But it has a lot of if you think about it in terms of machine learning, it's edge cases. So this is this is one of the things you have to think about when you model things in a stochastic sense. They make the point here, casinos heavily rely on the fact that although individual games are decided by chance, the odds are such that gamblers almost certainly lose in the long term. So the degree of randomness means that you can't really predict things in advance. You can predict things occasionally, but it's not something that when we do a biological experiment, for example, it wreaks havoc on our model because we can't make the kinds of predictions we're used to making, which are, you manipulate something, you get a certain outcome, and then it's repeatable. In a system like this, it isn't necessarily repeatable, and you have to account for that in your model system. So there are things like hidden variables as well. So hidden variables are variables that you don't identify a priori, but nevertheless have an effect on the system, on the outcome. And so there are models for uncovering hidden variables, hidden Markov models, but these are things that have to be, again, put into place in advance. So in self-ate choices, for example, these types of activities integrate a multitude of chemical and mechanical factors, which often vary with time and space. So a lot of these factors, and we've talked about them in previous meetings, we have to know what they all are. And of course, if we don't know what they all are, or don't know their full effect on the system, then we can't really say with any certainty what their effect is. So, we know that, for example, there's certain chemical factors or certain mechanical factors which predominate in shaping an outcome. So, we know that there might be forces that result in the differentiation of a cell, but those forces might have other mitigating factors, chemical factors, mechanical factors, maybe even genomic factors that all kind of fit together in that model of causality and explain the variance. So, we have to work with what we have. And if we think of the history of science has been where you start working on a problem and you only know maybe one or two causal factors or potential causal factors. And then as time goes on, you get other things that come into play that explain some of the variance in your result. So, you might have something that maybe explains 1% of the variance of what you're observing, of the outcome. But say you have things, when you start working on a problem, you might have things that account for 10 or 20 or 30% of the outcome. So, this process of sort of finding all the potential causal factors is, it's an interesting one to think about in light of reproducibility and indeed our understanding of differentiation as a phenomenon. So this is an example here where you have a probability distribution based on certain things, certain potential causal factors that exist. So if we just look at this outcome X, we see that there's this normal distribution of results. The value of X is normally distributed, so that means that in the center here at the mean, that's when most of your results, when you independent observations, will be in this category of the mean. But some of the observations will be on the tails, and so they're lower probability events, but they still occur. Now, if we look at, we know that, say, F1 has an effect on X, and we do an experiment where we look at those things, that sort of, the effect of F1 on X. So we might manipulate F1 and see the effect on X. Then we get a little bit different distribution. It's maybe skew normal, where you have these tails, but the tails are uneven, and the probability is maybe skewed towards the left here. So it skews once we know that F1 is involved and we manipulate F1, we revise our distribution and we say it's a skewed normal. And now, this is a Bayesian calculation here, X given F1. That's what the probability that we're looking at. So it's a conditional probability of X, but given that F1 is having an effect on it. But then we have, so we don't know about F2 and F3, although we can see in this hidden model that F2 and F3 have effects, and sometimes these are synergistic effects on each other. So now, we don't know about these in advance. We know that F1 is having an effect, but F2 is still having an effect on X, but we're not measuring that. So this is why our distribution is somewhat skewed. Now, if we go to where we know the full model, where we have F1, F2, and F3, and we know the relationships, and they're all observable, then we get a different outcome. We get this outcome where we measure the relationships here, and then now X is a different outcome, which is that most things occur on the mean, and very few things occur on the tail. And so this is an idealized model because my guess is that, there's a lot of slop in these measurements here because you don't know what these factors are upstream, and so it's very hard to really kind of get a good measure of X. But this is an idealized model, and it just kind of shows that as you uncover more of the mechanism, and you manipulate this, the effect shown and embodied by X changes over these different scenarios. But stochasticity is also, it's the experimental outcomes can appear more or less random depending on how much is known about the system. In mathematical terms, this can be illustrated using conditional probabilities. So they use conditional probabilities here, and it's just a matter of basically weighting X by the things that are affecting it. And so it's a sort of a more controlled way to measure X or look at X. And so this is why you get this change in probability distribution. This is all related to stochasticity because, for one thing, these variables may be stochastic. For another thing, you can see how sometimes you can get a better handle on the system. It becomes maybe more deterministic in the hands of the experimenter, or conversely, it could become less probable, or it could become more probabilistic and less deterministic, because now you have these variables which before you just assumed were not there. Now you know that there may be stochastic variables, and so they have a more direct effect on the system. So this can play out in a number of ways. So this is plugged into this model of self-renewal and differentiation. So sometimes we have cells in development that differentiate right away, and sometimes they differentiate later. And so why is that? Well, as it turns out, that developmental cells, or cells that exhibit meiosis, have a cell cycle, and that cell cycle goes in has a time period, and then at the end of that time period, the cell has to make a decision as to whether to self-renew and go through another cycle of meiosis or differentiate into a differentiated cell. And so this process of going through the cell cycle and then making this decision and then going through self-renewal, this is all can be modeled as a stochastic model. More specifically, it could be modeled as the roll of the dice, or the roll or the spinning of a roulette wheel. There's some, or maybe like some sort of stochastic switch where one state or another pops up depending on just kind of chance. And so this is all, like, you can put this in terms of casino games if you wish. I wrote a paper on cellular reprogramming and this sort of idea of a stochastic cycle where you have self-renewal or differentiation. And this has been addressed in the literature in a number of contexts as well. So this is definitely something to, if you're interested, there's a fair amount of literature out on this. So as these studies demonstrate, this can be leveraged to identify key factors that control cell-fate decisions, so we can combine the Bayesian model we saw in that figure that I just showed with this idea of self-renewal is sort of like a roulette wheel, where you have this decision that's made based on just kind of chance, or some chance outcome. And so as it turns out, though, those chance outcomes are modified quite a bit by other variables. So like I said, cells communicate with one another. Cells will, one cell might differentiate and then tell the neighboring cells to differentiate instead of renewing. So this can also affect the roulette wheel. It can put a thumb on the scale, as it were, and it can affect the outcome. So, your original variable driving this process is a totally stochastic process, but then you maybe have some deterministic cues that come into play that tell the cell to differentiate at a certain time. So we also have gene expression noise, and this is another area that's very important. Gene expression noise is something that occurs when genes get expressed, when the cell goes through the cycle and genes are expressed. It's not that the cells are completely without pattern. It's just that there's a certain amount of noise that's generated as genes get expressed. So there's noise in the amount of transcript that's produced. There's noise in the system that results in the expression of transcripts in RNA. And so this noise is a fluctuation of amount of this the amount of the molecule in the cell, or it could be just like fluctuations in time. So more gene product is produced at time A than time B, and then even more at time C. So there's this fluctuation. And so some people think that this noise actually does play a role in tuning some of these stochastic processes. So you have intrinsic sources of noise and extrinsic sources of noise. So intrinsic noise is sort of variation in that machinery. So when, transcripts in RNA are produced, they get produced at different rates over time, and there's fluctuations just based on like the different physiological conditions. There are also these extrinsic sources, which are environmental sources. So if the organism is under stress, or the organism is, got some other type of mechanism going on in the environment, it can create more noise and force the system into new states. So these molecular fluctuations are very important. And so they work together sometimes to influence differentiation of cells. Okay, so then they give another example from Drosophila nervous system. And this is a competence model versus a stochastic model. So there are different models of how these different things operate. Then they go into mammalian neocortex, and they talk a little bit about some of these things that happen in mice and in other types of organism. They go back to Drosophila, so they connect it back to Drosophila. So then they go into vertebrate neural retina, which is another good example of this. The retina is full of different types of cells. How do they differentiate? And then the idea that stochastic factors are also involved in retinoneurogenesis gathered traction when a study using dissociated retinal progenitor cells revealed that clones can vary highly in size and composition. And again, we've talked about this in C. elegans development, where cells have a certain size and composition in different tissues. This is even more the case in this sort of regulative development, where you have a lot of variation in the developmental program, as it were. And so this finding can be recapitulated using a simple stochastic model. So we can use stochastic models to find these different types of mechanisms, or to at least reproduce them. So they talk about dice here. They talk about throwing dice, and then the dice can be loaded so that certain outcomes are more likely than others. And so this self-fate decisions, when they're, when you're looking at a stochastic model, there are ways that you can look at it that say, there are influences on the stochastic. So it can be like a biased stochastic model. And so then they actually can do this in vivo lineage analysis. And they have this nice case where, one cell will produce one cell and one daughter cell, and then another daughter cell, which happens to be a die. So that's just representative of like the chance occurrence of a certain state at this node. So then conclusions is that this is the stochasticity is common throughout development, and that you can weight the coin or weight the die, as it were, in a stochastic process to favor one result over others. This is something that happens in nature, happens in tissues where you have multiple cells that influence one another, or things in the developmental program that override this stochastic process. So stochasticity is very useful for generating variation in development, but also a lot of these weighted factors actually keep it within the boundaries of what you might consider to be deterministic embryogenesis. Okay, one last thing here to finish up with is this. I was playing around with my Oculus Quest, and I found the OpenWorm Browser. And so I've been playing around with ways to render this OpenWorm Browser in the Oculus Quest visor. And so here's my Oculus Quest, and this is a live stream of what's going on inside the headset. So I'm wearing this headset, and this is my view. This is the web browser that comes with the Oculus. So this is not, this isn't a full screen version. I might try working on it to get it to work in full screen. But as you can see, my controllers are there, and I'm trying to manipulate the worm in the browser. So this is the browser that you find at browser.openworm.org. And you can see that you have a worm there that has, let me try the second one. You have a model worm that has different layers. You have the epithelial layer, which is the outer skin. You have the nervous system. You have the nerves, that are connect the neurons. You have the neurons themselves identified, and you also have some of the other organ systems. So you can see on the left there's this control panel where you can switch between those views. You can see in this example, these views we can show these views, and you can also manipulate the position of the worm. So this is a 3D model in Blender. If you look at this example, you can see that it's a standard Blender model, and it just needs to be brought into this virtual environment. It might actually end up building maybe a small app where we can render the worm and maybe fly through it. I don't really know if it's how this Blender model is about 10 years old, and we might be updating it soon. So stay tuned for more work on that. But this is one of our GitHub issues, which is building a virtual set of models for OpenWorm and DevoWorm, and more specifically. I guess I might want to, let's see what I want to do next.
Sorry for the interruption. I have to go. I have another meeting.
All right, well, thanks for attending.
I gotta go too. Yeah.
All right, take care.
Thank you very much.
Bye.
Read your book. Bye.
Yeah, bye.
Thank you, everybody.
Thank you.
Bye-bye.
Bye.